Hello, my name is Brandon Renshaw, and the unfortunate disease that I'll be talking about today is cystic fibrosis. So let's get into it. So, kind of an overview of the symptoms of cystic fibrosis, and the main one being thick mucus within the lungs that can harbor some chronic respiratory infections. And it starts at an early age. Those infections can leave scar tissue or fibrosis. That can also lead to cysts within the lungs as well. Um, that's kind of where the name of cystic fibrosis comes from, which is the fibrosis or scar tissue and the cysts that form within the lungs. Some other problems that can arise are digestive problems. Those typically happen in early childhood and progress through adulthood. And one of the main organs that have to do with that is the gut, so the intestines. And now there's typically mucus within the intestines that help to move things along. But having thick mucus from CF typically results in some digestive problems. Another organ that has to do with digestive problems is the pancreas. It secretes some digestive enzymes within the duodenum to help with breaking apart food and digesting everything. But the pancreas can also be affected in the release of insulin, which can lead to diabetes. And that's just because the thick mucus within the pancreas doesn't allow that insulin to reach the bloodstream very efficiently. Another problem that can arise is there can be, but not necessarily in every patient, there can be an absence of a vas deferens in males, and that vas deferens transports sperm from the testicles to the outside. So that male will be infertile, as it is a bilateral effect on most patients with that absence. So there's also fertility problems within females, and that's just because the endometrium typically is associated with having mucus, and therefore it's tougher to get pregnant that way. So I'm kind of going to start at the basics here. So there's chromatin within your nucleus of every single eukaryotic cell, which we are eukaryotes. Chromatin is in this picture at the top, in the light purple area, is kind of this squiggly line. It kind of just represents a mosaic of DNA where it's not really organized at all. But once that cell reaches the phase in which it's going to divide, and I'm going to kind of focus mainly on meiosis, that chromatin kind of organizes itself into what we would call chromosomes, and it's more known as this structure at the bottom here, kind of this X shape, right? So the part I really want to emphasize on this slide is what I just highlighted is chromosomes are made of DNA, right? And DNA really is made up of nucleotides. Now there's two strands. There's one strand, and then it's complementary strand, and that's made up of nucleotides that are paired up, and that's what we call base pairs. As we see here, this A is paired with a T, T with an A, G with a C, A with a T, and C with a G. And those nucleotides are adenine, thymine, guanine, and cysteine. And so that creates your double helix. Your double helix kind of wraps itself around some histones, this little protein up here. Those histones become nucleosomes in kind of grouping together. And that kind of becomes more tightly wound into chromatin and therefore into this X-shaped chromosome here. Now that chromosome, there's obviously names because science. So there's some names that are associated with a chromosome. The first of which is that the chromosome typically is made of these two strands binded at the center here. And these two strands are called chromatids. and they are connected here with a centromere, represented by this little red dot. And then there's a P arm up at the top, number 3, and that's your short arm. This Q arm is kind of the long one down at the bottom here. And going into some chromosome action within meiosis, so there's 2 steps and 4 phases per step. And so within the first step, that's prophase. That's kind of when your chromatin, that little mosaic, organizes itself into actual chromatids, chromosomes, as we would traditionally see. So those homologous pairs, since we are diploid cells, we have pairs of chromosomes. We don't just have 46 separate chromosomes. We have 23 pairs of chromosomes. So those homologous chromosomes kind of line up along the center here in metaphase, and then they are pulled apart in anaphase, and then that cell actually divides via telophase and cytokinesis. And the main point in that is this cell over here had 46 chromosomes, and these 2 resulting cells have 23 chromosomes, and that makes those cells haploids. And this is creating gametes, so either sperm or ovum. And how we get back to being a 46 chromosome diploid creature, which we are, those 2, sperm and an ovum, have to fuse and within a mating session, and that creates your 46 chromosome diploid creature. The main point I really want to get across on this slide is genes. So those genes that were going through, so those cells that were going through meiosis kind of had some action within them, and that action really is crossing over. So let's say this B mixed with this B, and they just kind of crossed over, and those are on homologous chromosomes. Let's say chromosome 7, since we're going to get into depth on that one today. So the main point on this one is that there's a difference between homozygous and heterozygous, as you can see on this picture. There are dominant genes and there's recessive genes. If you have 2 dominant genes, you're homozygous for a dominant gene. And if you have 2 recessive genes, then you are homozygous for that recessive gene. And if you have a mixture of the 2, then you are heterozygous. And that dominant gene will be expressed over that recessive gene. And so you're going to show whatever that dominant gene is, unless there's no dominant gene at all, in which case you're homozygous for this recessive gene, and therefore you will show that recessive gene trait. And the reason I bring that up is because a mutation within the CTFR gene typically is a recessive gene, and if you are homozygous for that recessive gene, then you will have cystic fibrosis. So getting into the CFTR gene, which stands for cystic fibrosis transmembrane conductance regulator, there will be a test. But I'm going to call it CTFR. Yeah, it's easier. But that CTFR gene is located on chromosome 7, which is highlighted in red over here. I kind of circled the general area where that CFTR gene is located. Its actual position is Q31.2. That Q is representing that this is on the Q arm, which is that long arm at the bottom. So within genes, there's mutations. Mutations is kind of an inevitable fact of life. Everybody will get them. But to kind of go over the mutations as a whole, mutations can come in many different flavors. There can be a deletion of a nucleotide, there can be an insertion of a nucleotide, which changes the genetic sequence, or there can be a replacement of a nucleotide, which again changes the genetic sequence. But any mutation really is just a mistake within the sequence that leads to abnormal proteins via the transcription and translation of into proteins. Speaking of proteins, that CFTR gene will translate itself into the CFTR protein channel. Now that protein channel is located on our cell walls, right? So and that protein facilitates the movement of chloride ions into and out of the cell, and water will follow those chloride ions via osmosis. And thick mucus is really just a byproduct of not having enough water to make it kind of what it's for, which is a surfactant. And so that's where you get your thick mucus from, is this lack of chloride ions and the resulting water following it. A consequence of that is having this thick mucus, as you can see on the cell on the right, this structure on the right, which is your trachea. You have some inflammation of this muscle. You have an increased mucus secretion, and that mucus is going to be, again, pretty thick, resulting in different chronic infections and any number of symptoms that we went over before. So kind of a pedigree of how you would wind up with cystic fibrosis. Let's say that you are, in this chart, you're this little girl over here, and you have cystic fibrosis. Well, if you do, that just means either that you have a carrier for a mother and a carrier for a father, which just means that they carry that recessive gene. Or you can also have a mother that has cystic fibrosis, which means you would have two recessive genes, but your father would also have to be a carrier, and then you would wind up with two recessive genes. And a good representation of that is this Mendelian model here, or a Punnett square, as we like to call them. But this is just representative of the pedigree that we see on the left. Your mother is heterozygous, and so is your father, and therefore the resulting offspring will have a 25% chance of winding up with cystic fibrosis. The other results of a mating is 50% will wind up as carriers, and another 25% will wind up as unaffected. They don't have that recessive gene. But the main point is that carriers, if a carrier mates with another carrier, they have a 25% chance of having a child with cystic fibrosis, and a 75% chance that cystic fibrosis is not going to be present. But those children could be carriers as well. So kind of on to genetic testing. The testing for cystic fibrosis is pretty much nationwide. Every state has a protocol of screening for CF, and some are more effective than others. However, there will have to be subsequent testing to confirm or deny actual results. And there are some tests for parents if they're thinking about having children, to test to see if they are carriers. And from that, you can use a resulting Punnett square to find out what the percentage is that you might have a cystic fibrosis affected child. Anyway, that was my presentation. I appreciate you listening. Thank you.